On this week's GARNet Research Roundup, I'm delighted to be joined by Martin van Rongen, who works with Ottoline Leyser at the Sainsbury Lab in Cambridge. And we're talking about a paper just published in PLos Genetics, and it truly is a genetics tour de force. The title of the paper is Connective auxin transport contributes to strigolactone-mediated shoot branching control independent of the transcription factor BCR1. So there's a lot in the paper to unpick, so welcome, Martin. It would be great if you could give us an overview of what you achieved in this work.
Yeah, my pleasure. So the aim of this paper was to investigate in more detail the role of auxin transport in the regulation of shoot branching. And our lab has previously shown that strigolactone affects the auxin transport network, which in turn influences the bud activation process. And we know that this effect mainly happens in the auxin transport stream in the stem, which acts as a sort of highway for auxin transport that is produced at the shoot apex and then moves it towards the root. And the way we view it is that in a plant, all the buds along the stem kind of feed into this transport stream, and they have to establish their own auxin transport in order to become and to remain active. And most of the previous research has focused on PIN1, which is is one of the most important auxin export proteins in Arabidopsis, for example. And the problem with PIN1 is that that if you lose it, that it makes the plant sterile and also reduces the number of buds that are formed. So from a genetic point of view, it makes it rather difficult to study this kind of mutation. Mm-hmm. However, a couple of years ago, we found that closely related proteins, namely PIN3, 4, and 7, contribute to a more widespread auxin transport in the stem, which we called connective auxin transport. And we called it this way because it allows two-way communication between the auxin transport stream and the surrounding tissues in the stem. Mm-hmm. And we found that it also affects communication between buds. But if and how connective auxin transport is also affecting strigolactone-mediated branching wasn't really clear yet, and that's what the current paper then focuses on. And in this paper, we show that connective auxin transport actually does impact on strigolactone-mediated branching, because if you lose PIN347, it results in a partial rescue of the increased branching that you see in strigolactone mutants. Mm-hmm. We not only focus on PINs, but we also look at ABCB19, which is another auxin export protein, which is also broadly expressed in the stem. And we show that ABCB19 is also able to the branching in both wild type and strigolactone mutants. So there's some clear similarities between the effects of PIN347 and ABCB19 in terms of shoot branching, but actually what I find the most exciting finding of this paper is that there are also some very clear differences between ABCB19 and PIN347. Most of that impacts on another part of the bud activation network, because we know that strigolactone also regulates the expression of a transcription factor called branched1 or BRC1, mm-hmm. and plants that lack BRC1 are also very branchy. And interestingly, if you lose PIN347 in that kind of mutant background, it doesn't do anything to the branching, but if you lose ABCB19, it does. So, and that's kind of where the paper ends at the moment, and we'll see how to develop it from there.
Okay, sounds like you've given us a clue as to what might be coming down the line. But so it's interesting. So the key concept of this, one of the key concepts in the paper, is this idea of the connective auxin transport, which kind of moves auxin across the stem instead of up and down as with polar auxin transport. So you mentioned there about this triple mutant, the PIN347, but do you see any effects in the individual mutants or the double mutants, or do you need to knock out all three of them to have any effect here?
So we touched a little bit upon that in the previous paper that we published in 2016, where we looked at the effect of single and double mutants as well. And we look into that a little bit in this paper as well, but mostly the strongest effect you see when you lose all three. So we think that there's some kind of redundancy between these pins. Okay. So if you lose only just one or two, then the other one will be able to compensate for that. Although some of the phenotypes that we see in the triple mutant, you see to a lesser extent in some of the double mutant combinations as well.
Okay, so that's interesting. So, I mean, this isn't the focus of this paper, but we know from previous work in the roots that the PIN3 and the PIN4 are in different cell files. Is that what you see in the shoot as well? Are they in different cell files, or do they overlap their expression their expression patterns?
A bit of both. So,
Okay.
There's definitely overlap within the stem. What you tend to see is that, for example, pin 3 has quite a broad domain.
Okay.
It has more of a polar localization than, for example, pin 4, whereas
Okay.
pin 7 has kind of the broadest domain of the three of them, and is a lot less polar than the other two. So it's a difference in expression domain, but also in the polarization that you see.
Okay, so you mentioned about the roles of the pins, and then also this ABCB19 protein as well. So can you give a little more detail about how these differently act?
Yeah, so I can. So one of the things that what we found before is that strigolactone is able to affect the localization of pin 1. So if you add strigolactone to pin 1, then what happens is that you end up with endocytosis. And that the amount of endocytosis that takes place has an effect on how much transport there is, which then affects bud outgrowth. So what we try to see is whether or not, for example, in strigolactone mutants, you have differential localization or expression of these pins that we look at now, so pin 3, 4, and 7. We didn't see a huge amount of effect, only a slight increase in pin 7. But not the others. But then we also looked at ABCB19 to see if that is differentially expressed. So we looked in a D14 strigolactone receptor mutant, and quite surprisingly in the stem there's absolutely no ABCB19 that we can detect, whilst in wild type there's a lot of it across all the tissues in the stem. So how that directly relates to the shoot branching, we're not quite clear. So we'll have to look in other tissues as well.
Kind of the final part of the paper, you make actually you do a quintuple mutant, right? I think of the BCR 1 and 2, and then the pins as well. So it's always impressive to see these higher order mutants be made. So can you give us a quick overview about the role of BCR1?
Yeah, so generally what branch 1 is regarded as a kind of integrator of various signals, and then the amount of branched 1 that is present in the bud then determines whether or not a bud is going to grow out or not. So if you have a lot of branched 1, a bud will remain dormant. If you have very little branched 1, then a bud will be able to release from dormancy. And we've also shown that in other work that that correlation between branched 1 amount and the activity of buds can actually be broken, because, for example, branch 1, 2 mutant plants don't activate all their buds in some circumstances, and that's what we kind of continue on with here. So if you grow plants for a long time, they form many more buds than when you grow them in, for example, these normal long day conditions. Okay. But if you grow them for longer, they have more buds, and then you start to see these more subtle effects. So that's what we did in this paper as well. And then we asked whether or not losing pin 3, 4, 7 would be able to further reduce the branching that you see in these branch 1, branch 2 double mutants. And we didn't see any effect in any of the conditions that we tested. Okay. And the difference there is that when you lose ABCB19 in the branch 1, branch 2 mutant background, you do see some effect, where there's some reduction in branching, and that's what we try to come up with some kind of explanation for that. So.
Yeah, I see. So, I mean, that kind of gives a clue. I always like to ask about, future work, where the work goes after this. So can you give us a little teaser about some stuff which you might coming out later on, and maybe it sounds like they might be about the interaction between the pins and the ABCB, perhaps. So.
Yeah, so in terms of interaction between the pins and the ABCB19, not so sure, because we touch on that a little bit as well, where we made a ABCB19 pin 3, 4, 7 quadruple. You see some additive effects, and mainly in terms of shoot branching, you see some interesting aspects that if you don't have ABCB19, bud activation following decapitation is rather rapid. Whereas if you lose pin 3, 4, 7, then it's kind of slowed down and then kind of catches up towards the end. And that kind of gave us a bit of a hint of what might be going on. And what we think is that perhaps ABCB19 has a role in loading auxin at the bud apex, because previous research by Angus Murphy's lab has shown that ABCB19 at the normal shoot apex is important to load auxin into the polar auxin transport stream. And what we're wondering is whether or not it might do something similar in the bud.
Okay.
And that is definitely something that we would like to follow up on, because it would kind of tie in nicely with the results that we found here and would provide an explanation for them.
Okay, cool. All right, well, thank you very much, Martin, for chatting to us today. I mean, as ever, I urge people to go and have a look at the paper. It's somewhat complicated. There's a lot of genes involved and a lot of mutants made. So thanks very much for joining us today and discussing the paper.
Thank you.